both of each kind of touched on what your approaches are. Could you maybe give, for the benefit of those who can't be here tomorrow, kind of in a nutshell description of the clinical trials you're currently doing, and if those pan out, what you think your next steps might be?
So there are cure clinical trials taking place right now. It goes beyond theory. Not all that they're talking about is theoretical. We don't know outcomes like they're talking about, but there are trials happening on the ground right now. So
I can do mine real quickly. So I've been involved with a clinical trial that's going to be sponsored by City of Hope Hospital in Los Angeles, and that's going to be using zinc finger nucleases to cut out the CCR5 gene. So very similar to clinical trials that have already taken place and are ongoing. The difference is that the previous clinical trials have been taking people's T cells out of their body and engineering them with the zinc finger nuclease. And the trial that I'm involved with is actually going to be taking people's bone marrow stem cells out and doing the same treatment. The potential advantage of that is because the bone marrow stem cells are actually the cells in your body that produce your CD4 T cells. It's like you're kind of going back in time and engineering the mother of the CD4 T cells. And so if it works, it's potentially a longer lasting treatment. So that clinical trial is now up and available to look at on clinicaltrials.gov, one of my favorite websites. And it's sponsored by City of Hope. And I believe we're at the stage of, we have several potential patients under evaluation who are being kind of worked up and looked to see if they qualify to go into the trial.
I'm not going to talk about any ongoing clinical trials tomorrow at all. I'm going to talk about the what we call VIP, which is vectored immunoprophylaxis. It's a
that's
not exactly
just runs off the tongue,
right,
doesn't it?
Right. That's why we call it VIP. And the rationale behind that, and the which is to use genes that encode highly broadly active, highly active antibodies to protect, as I said, humanized mice and in one experiment, monkeys. And we're soon to get that into people, but haven't yet.
So are you doing that? Is that trial going to happen in England? Did I read that right?
No. Phil Johnson has a trial which is going on in England at the moment, which is very similar to what we want to do. We're using a different antibody, and I don't know any results from that. There is no public information about that. Ours is going to be in the United States.
Okay. So David's approach, I'm going to pitch it for him. It's actually a really cool idea because there's many reasons why vaccines just aren't working against HIV. And I suppose the simplest way to think about it is when you are HIV infected, your immune system knows you're HIV infected and it starts to make antibodies against the virus. And if you were infected with something like the common cold, you know what it's like. You get it for 7 to 10 days, and then your antibodies and your immune system kind of get the upper hand, and they cure you of your flu virus. And that doesn't happen with HIV. And part of the problem is, HIV is just very good at disguising itself from our antibodies, and it's constantly changing. So because even people who are infected with HIV, even, which is, if you like, the best case scenario when it comes to making antibodies, even their antibodies don't protect them from HIV. That goes some of the way to explaining why we can't give people a vaccine to make antibodies that will protect them from HIV. David's approach, what it's done is it's almost like it's kind of scanned the whole world and looked at all the antibodies that everybody makes, and it's found kind of like sort of cherry-picked.
The super antibodies.
Yeah, the super antibodies that some people have made that just really work really well and work against lots and lots of different HIV, no matter how variable HIV tries to be. And they said, okay, this is like the gold standard antibody. Now, can we make people make those antibodies? Can we inject them with something that will make them make those antibodies? No, they haven't figured out how to do that. So instead they say, okay, let's just take these antibodies and let's do something that never happens in nature. Because this is what gene therapists do all the time, let's take that actual antibody and give people, using gene therapy, give them that antibody. It's like kind of like a, a la carte menu option. Let's have them make that antibody. Rather than have them try and make it themselves, we're just going to actually give them a kind of a ready-formed antibody. So this is what David's sort of talking about. So again, it's a radically different way of thinking about treating a disease. But it comes back to this idea of, the more we know about how our immune system works, the more as smart, Homo sapiens, we can think about ways to kind of improve our immune system and actually, do things that are, quite preemptive. So I think it's a very radical approach.
We actually program muscle cells to make the antibody.
Yeah.
Now, muscle cells don't usually make antibody at all. And so we've gone completely around the immune system.
Why did you choose muscle cells?
They chose us. I'll show you tomorrow.
They're also easy. They're also easy to get at. It's easy to sort of give people
It is.
in the muscles. Yeah.
Back in the old days, you used to get vaccines by turning over and getting a shot in the tuchus. And
wait, the shot in the what?
It's a New York word.
You need a lesson in Yiddish. And that's because there's a nice big muscle back there, that gluteus, right?